Nipah virus, NIV, is a highly lethal zoonotic pathogen first identified in the late 1990s. Its history is marked by a shift in transmission dynamics from pig to human in early outbreaks to direct bat to human and human to human transmission in more recent ones. Key historical milestones: discovery in Malaysia 1998-1999. The virus was first identified during a major outbreak among pig farmers in Kampung Sungai Nipah, Malaysia. Initially mistaken for Japanese encephalitis, the outbreak infected 265 people and killed 105. To contain it, over 1 million pigs were culled. Spread to Singapore, 1999. An outbreak occurred among 11 abattoir workers who had contact with pigs imported from Malaysia. Emergence in Bangladesh and India, 2001-present. Bangladesh 2001, first reported in the Meherpur district. Unlike Malaysia, outbreaks in Bangladesh occur almost annually and are often linked to drinking raw date palm sap contaminated by fruit bats. India, 2001, 2007, 2018 to 2026. Early outbreaks occurred in West Bengal, Siliguri, and Nadia. Since 2018, Kerala has become a recurrent hotspot. Philippines outbreak, 2014. A unique outbreak was linked to the slaughter and consumption of horse meat, marking a new intermediate host for the virus. Evolution of transmission and strains. Researchers have identified two distinct genetic lineages. NIV Malaysia, Nivinum, primarily transmitted via pigs, associated with a lower, though still high, fatality rate of approximately 40%. NIV Bangladesh, NIVB, associated with direct bat-to-human spillover and frequent human-to-human transmission. It typically has a higher fatality rate, often exceeding 70%. Recent outbreak, January 2026. In January 2026, Indian health authorities confirmed a new outbreak in West Bengal, including cases among healthcare workers. This led to heightened airport screenings across Asia, including in Thailand and Vietnam. World Health Organization, WHO status due to its high mortality rate 40% to 75%. And lack of available vaccines or treatments, the WHO priority pathogen list includes Nipah as a disease with epidemic and pandemic potential. Building on its discovery, the history of Nipah virus, NIV, has transitioned into a period of intensive vaccine research and refined outbreak management, driven by its potential to cause a global health emergency. In January 2026, a new cluster was identified in West Bengal, India, marking the first occurrence in that state in 19 years. 2 healthcare workers, nurses in Barasat, were confirmed infected via RT-PCR testing on January 13, 2026. Health authorities traced and tested 196 contacts, all of whom tested negative, suggesting successful containment of this specific cluster. The event triggered airport screenings in Thailand, Nepal, and Malaysia for travelers arriving from affected regions. Building on the early history, the landscape of Nipah virus, NIV, research and global response has reached critical turning points as of early 2026. While it remains a high mortality threat, significant breakthroughs in medical countermeasures are finally entering late-stage testing. After years of being a priority pathogen with no available prevention, several candidates have reached human trials. Oxford CHAdOx 1 Nipah. In December 2025, the University of Oxford launched the world's first Phase 2 clinical trial in Bangladesh. This vaccine uses the same viral vector platform as the AstraZeneca COVID-19 vaccine. Tokyo University Vaccine. Researchers developed a candidate based on the measles virus platform. Following successful animal trials, Phase 1 human trials are scheduled for April 2026 in Belgium. Self-amplifying mRNA. Genova Biopharmaceuticals is developing an mRNA-based candidate with funding from the Coalition for Epidemic Preparedness Innovations, CEPI. In addition to the geographic spread and vaccine updates, the history of Nipah virus is significantly defined by the long-term challenges faced by survivors and the complex ecological patterns of its natural reservoir. While many survivors of the acute phase recover, roughly 20% of survivors experience lasting neurological or psychiatric conditions. These sequelae can persist for years and include persistent symptoms, frequent reports of debilitating fatigue, daytime somnolence, and recurrent seizures. Neuropsychiatric changes. Survivors often face cognitive impairment, memory loss, depression, and significant personality changes. Relapsing encephalitis. Uniquely, Nipah can cause late onset or relapse encephalitis, where survivors fall ill again months or even years after their initial recovery due to the virus, remaining dormant in the central nervous system. To wrap up the clinical and diagnostic history of Nipah virus, recent efforts have shifted toward addressing the long-term consequences for survivors and the high risk of misdiagnosis. The hidden burden, post-Nipah syndrome, as more people survive outbreaks due to improved supportive care, a pattern of long-term neurological issues has emerged. Neuropsychiatric effects. Survivors frequently report persistent fatigue, personality changes, depression, and cognitive deficits, making long-term rehabilitation a critical part of modern outbreak management. Diagnostic evolution because early symptoms, fever, headache, cough, mimic common illnesses like influenza or malaria. Rapid testing is the new historical frontier. RT-PCR. This remains the gold standard for detecting the virus in throat swabs, urine, or blood during the acute phase. ELISA testing, used later in the illness to detect antibodies, e.g. and IgM, helping confirm the diagnosis even after the patient has recovered or passed away. Point-of-care breakthroughs. Research is currently focused on developing rapid diagnostic kits that can be used in rural clinics in Bangladesh and India, where laboratory access is limited. Pandemic preparedness, 2026 and beyond. The WHO R&D blueprint continues to list Nipah as a priority. Because of its high mutation potential and the mobility of its natural host, the Pteropus bat. Surveillance. Monitoring bat migration patterns has become a year-round activity in Southeast Asia to provide early warnings for spillover seasons, typically December-May. Global stockpiling. Discussions are underway regarding the creation of a global stockpile of monoclonal antibodies to be deployed immediately when a new cluster is identified. The modern history of Nipah virus concludes with its current status as a benchmark for global pandemic preparedness. Following the January 2026 West Bengal cluster, health authorities have refined diagnostic and isolation protocols. Rapid diagnostics. The WHO has highlighted the use of real-time PCR to confirm cases within 24 to 48 hours, a massive leap from the weeks-long delays seen in the 1990s. Contact management. Of the 196 contacts monitored during the 2026 outbreak, a rigorous 21-day quarantine was enforced, reflecting the upper limit of the virus's incubation period. The One Health milestone. The history of NIV has forced a shift from reactive medicine to proactive ecology. Bat monitoring. Countries like Thailand and Malaysia now utilize long-term surveillance of Pteropus bats to detect viral shedding spikes before human spillover occurs. Environmental policy. Governments in South Asia are increasingly regulating the date palm sap industry, providing bamboo skirts to farmers to prevent bat contamination. Future outlook. 2026 and beyond. The goal of the Coalition for Epidemic Preparedness Innovations, CEPI, is to have an approved vaccine or a warm manufacturing base ready by the end of the decade. This would move Nipah from a neglected tropical disease to a manageable threat.